A frozen needle pierces an Asian elephant egg in Boston. Cold glass shimmers under white fluorescent light. Deep inside the microscopic chamber, geneticists insert 59 ancient genes recovered from frozen hair into living Asian elephant DNA. The heartbeat begins. Can 40,000 years of subterranean silence be broken by a single modified cell? Your body holds an ancient memory. Your ancestors stood upon icy windswept tundras 15,000 years ago. They tracked massive herds across frozen valleys while shivering inside stitched animal hides under a pale prehistoric sun. The earth trembled beneath heavy feet. Why did those majestic beasts vanish from your planet so abruptly? Vast grasslands once carpeted the northern hemisphere. Golden steppe grass stretched continuously from Western Europe across Eurasia into North America. During glacial peaks, immense herds of woolly mammoths, steppe bison, and wild horses grazed across millions of square miles under clear freezing skies. Prehistoric hunters followed their movements. How did your ancestors survive alongside such overwhelming megafaunal abundance? Scientists found a mummified calf named Yucca and her thick auburn fur remained intact. Soft muscle tissue and preserved blood vessels survived inside the permafrost, deep soil that has remained frozen solid for thousands of consecutive years. Dark marrow leaked from cracked bone. Could living tissue endure inside frozen darkness forever? Archaeologists excavated ancient ivory shelters at the Kostenki site in Russia. Your ancestors constructed massive winter huts using stacked mammoth skulls and interlocking ribs. They burned heavy mammoth fat inside stone hearths to survive bitter glacial nights. Smoke drifted into freezing skies. Did human hunting pressure drive the final megafauna over the edge of extinction? Harvard geneticist George Church entered the Siberian tundra. He targeted specific physical traits. Using CRISPR gene editing, a molecular pair of scissors that cuts and edits precise sequences of DNA, his team spliced mammoth cold adaptation genes into elephant cells. Dense fat layers emerged. Small ears evolved to prevent frost. Would a hybrid embryo survive inside a living surrogate mother? Scientists modified metabolic pathways inside living cells. They altered lipid production to withstand sub-zero temperature drops. Cold-tolerant blood proteins were synthesized inside laboratory flasks to transport oxygen through freezing veins. Microscopic needles twitched under blue light. Can modern biotechnology resurrect a lost circulatory system? Paleogeneticists extract microscopic bone dust inside clean rooms. Ultra-pure chemical solvents dissolve calcified mineral matrices. Next-generation sequencing machines read billions of short, damaged DNA fragments to reconstruct a digital map of the extinct mammoth genome. Computers align scattered genetic codes. Can digital algorithms accurately reconstruct a living blueprint from shattered molecules? Paleogeneticist Beth Shapiro raised a crucial warning. Exact cloning remains impossible. Ancient DNA degradation, the natural fragmentation and chemical rotting of genetic strands over thousands of years, leave scientists with incomplete biological code. They can only create a proxy species, an animal engineered with modified genes to mimic an extinct creature. Is an engineered elephant truly a woolly mammoth? Evolutionary geneticist Love Delaine extracted ancient bone fragments from Wrangel Island. That lonely Arctic sanctuary held the final living mammoth herd until 4,000 years ago. Isolated populations accumulated harmful genetic mutations leading to lost olfactory genes, damaged coats, and incurable diabetes before final extinction. Inbreeding shattered their biological resilience. What if resurrecting an extinct giant recreates its terminal genetic suffering? De-extinction, the scientific process of resurrecting extinct species through genetic engineering, is not a zoo exhibit. The true target lies buried beneath Arctic ice. During the Pleistocene, the ancient epoch of ice ages that ended 11,700 years ago, millions of heavy mammoths trampled the vast northern tundra. Grasslands flourished under heavy footprints. Did you know that bringing back the mammoth is actually an attempt to stop a global climate catastrophe? Geophysicist Sergei Zimai of Pleistocene Park in Siberia. He measured deep ground temperatures. Without giant herbivores crushing winter snowpack, insulating snow keeps the underlying permafrost warm enough to melt. Decaying organic matter releases subterranean methane. What happens to your city when trillions of tons of greenhouse gases erupt from Siberian mud? Herds of engineered mammoths must scrape away winter snow. Their massive tusks expose soil to freezing Arctic air. Deep permafrost refreezes solid under intense cold, trapping ancient carbon beneath impenetrable sheets of blue ice. Grassroots stabilize thawing earth. Can an artificial herd save your modern coastline from rising seas? Tundra vegetation requires constant physical disturbance to thrive. Deep roots absorb excess moisture from thawing topsoil during short Arctic summers. Heavy hooves break up dead moss layers to allow nutrient-rich grasses to spread across vast plains. Soil microbes slow their metabolic activity under cold grass cover. Will resurrecting ancient grazers restore the lost steppe ecosystem? Asian elephants communicate through subterranean infrasound vibrations. Low-frequency rumbles travel miles through solid earth. Female matriarchs process ground vibrations through sensitive nerve endings inside their feet and trunks to coordinate herd movements. Engineered proxy mammoths inherit those same acoustic traits. Will synthetic herds detect distant warnings vibrating through frozen permafrost? Surrogate elephant mothers face immense biological strain. Artificial womb technology remains unproven. Engineered calves must learn complex herd behaviors, seasonal migration paths, and survival tactics without living wild mothers to guide their steps. Solitary calves wander freezing pastures. Who will teach a synthetic orphan how to survive an Arctic blizzard? Epigenetics, chemical tags on DNA that turn specific genes on or off, depends heavily on maternal environment. A surrogate Asian elephant mother provides warm blood. Yet the engineered embryo carries instructions forged in sub-zero winds, creating a cellular conflict inside the womb. Tension builds inside living tissue. Will the surrogate body reject the icy visitor within? Young mammoths required extensive matriarchal social structures. Older females transmitted knowledge about hidden water holes and seasonal migration routes through low-frequency rumbles. Synthetic herds created in laboratories lack experienced matriarchs to guide their journey. Young calves stumble through deep snow alone. Can instinct overcome the absence of ancient cultural memory? Conservationists question the immense financial investment required for de-extinction. Millions of dollars flow into private biotechnology ventures. Meanwhile, modern Asian elephants face severe habitat loss, human conflict, and poaching threats across their remaining tropical sanctuaries. Living species slide toward extinction today. Is resurrecting past ghosts worth the risk of neglecting living giants? Your ancestors painted curved tusks upon the walls of Chauvet Cave 30,000 years ago. They rendered thick auburn coats using crushed charcoal and mineral pigments under flickering torchlight. Those ancient artists captured living giants with reverence and deep fear. Today digital codes replace charcoal lines on stone. Has human hubris finally conquered natural extinction? Your lungs breathe air shaped by ancient ecological shifts. Your future rests upon decisions made in genetic laboratories today. The return of the mammoth forces you to choose between preserving remaining wilderness or re-engineering shattered ecosystems from raw code. Will your generation allow an ancient needle to thaw? A living calf steps into the snow and a frozen needle melts forever.